Hello everyone, welcome to my YouTube channel. So in this video we will discuss on the topic glycolysis. As the name suggests, lysis means here something is breaking down. Here ultimately the glucose molecule that we intake through our food is breaking down into pyruvate, ATP and NADH to provide the cell with energy. This pathway was discovered by Emden, Meyerhoff and Parnas. So this pathway is also called as the EMP pathway. It is an oxygen independent pathway, that is, it does not require oxygen for the pathway to occur. It takes place inside the cell cytoplasm. Cell का जो cytoplasm है वहाँ पे glycolysis process is carried out. Glycolysis process we can say that glycolysis process is the first step of cellular respiration. Cellular respiration का first step है, जैसे कि final जो oxidative phosphorylation जो है वो final step है cellular respiration का, वैसे glycolysis क्या है? It is the first step of cellular respiration. Glycolysis involves 10-step reaction. It is a 10-step reaction into which various enzymes are involved, and ultimately the glucose molecule, one molecule of glucose will ultimately give rise to 2 molecule of pyruvate, 2 ATP molecule and 2 NADP. NADP can also be converted into ATP. So ultimately it provides the cell with energy in the form of ATP. So let's see the steps of glycolysis. So this is the step of glycolysis process. First, when we take food that contains glucose. So glucose is a 6-carbon molecule. So glucose will convert into glucose 6-phosphate by the presence of enzyme hexokinase. Hexokinase is an enzyme that is present in the liver cells. Then glucose 6-phosphate will convert into fructose 6-phosphate. Here isomerization occurs and the enzyme that is involved is isomerase enzyme. Fructose 6-phosphate will convert into fructose 1,6-bisphosphate. This is also 6-carbon compound, and the enzyme that is required for the conversion of fructose 6 to fructose 1,6, that is, the enzyme is called the phosphofructokinase. That is also called as the PFK. Then fructose 6-phosphate, as it is a 6-carbon molecule, it divides into 2 compounds, each having 3 carbon. First is glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate is a substance that we require, and another 3-carbon compound it will get converted, that is dihydroxyacetone phosphate, that is DHAP. DHAP can convert into glyceraldehyde 3-phosphate by the presence of enzyme triose phosphate isomerase. So ultimately DHAP will convert into glyceraldehyde, and glyceraldehyde can also convert into DHAP. So this is a reversible reaction in which DHAP can convert into glyceraldehyde, glyceraldehyde can also convert into DHAP. So as DHAP will convert into glyceraldehyde, ultimately it will form 2 molecules of glyceraldehyde. 2 molecules of glyceraldehyde 3-phosphate will be formed. After that, the reaction will continue from 2 molecules of glyceraldehyde 3-phosphate. Then after the glyceraldehyde 3-phosphate is formed, when 2 molecule of glyceraldehyde is formed now, because the DHAP also forms glyceraldehyde 3-phosphate, so here 2 molecules of glyceraldehyde is formed. Glyceraldehyde then again it converts into 1,3-bisphosphoglycerate by the presence of enzyme glyceraldehyde 3-phosphodehydrogenase. Then 1,3-bis, in this reaction, in this conversion, when glyceraldehyde will convert into 1,3-bisphospho, so here 2 molecules of NADH is released. We know that NADH can also be converted into ATP, so that we will understand later on. Here 2 molecules of NADH is released. Then when 1,3-bisphosphoglycerate convert into 3-phosphoglycerate, here also 2 molecules of ATP is released. Why we are taking 2 molecule? Because we have 2 molecule of glyceraldehyde 3-phosphate. Here at this place we have 2 molecule of glyceraldehyde 3-phosphate. So as we have 2 molecules, so everything we will multiply it with 2. If it is releasing then also we will multiply it with 2. If it is using something, then also we will multiply it with 2. Then comes 3, from 3 phosphoglycerate it will convert into 2 phosphoglycerate. The enzymes are all the enzymes that are involved I have written in pink color. So all the enzymes are pink color which are involved in each and every steps. Then 2 phosphoglycerate will convert into phosphoenol pyruvate. At this step, enolase enzyme will be involved and 2 molecules of H2O will be released over here. Here not one, here 2 molecules of H2O will be released. Then after the process phosphoenol pyruvate, it will convert into pyruvate. Ultimately what we want in the glycolysis process, the end product is the pyruvate. Pyruvate, here also 2 ATP is released and the enzyme that is involved is pyruvate dehydrogenase. So this was the 10-step reaction of glycolysis. Ultimately the end product that we get from glycolysis is the pyruvate. So pyruvate is either, it depends कि if we are using oxygen or मतलब कि if it is taking place in an aerobic organism or in an anaerobic respiration pathway. So if it is occurs in aerobic respiration, then the pyruvate that is formed will have another fate as compared when it occurs without the oxygen. Suppose the pyruvate that is formed occurs through aerobic respiration. So in this pathway, in this method, in this way, if the pyruvate is formed by using oxygen, the pyruvate will enter into Krebs cycle and it will undergo oxidative phosphorylation and it will lead to the production of 32 molecules of ATP. If it is using oxygen, if it is occurring in an aerobic respiration, the pyruvate will enter into the Krebs cycle, which we call the tricarboxylic acid cycle or citric acid cycle, the TCA cycle that will enter, and ultimately it will give rise to 32 molecules of ATP. If the pyruvate is occurring in an anaerobic organism, so if it occur in an anaerobic respiration process, then it will it won't enter into the TCA cycle. Now here the pyruvate will convert into lactate. It will remain in the cytoplasm and it will convert into lactate and it will give 2 molecules of ATP. Here it will give 32 molecules. Here it will give 2 molecules of ATP. So this was all about the glycolysis pathway. And remember always one thing, that the step 1, 3 and 10. These 3 steps of reaction, 1, 3 and 10. In this step, it is an irreversible step. Okay, and step 7 and step 10 is a substrate level phosphorylation. Means it here in step 10 and step 7, here ATP is given out. ATP जो लास्ट में है, जो 2 molecule total 4 molecule of ATP is given out and total 2 molecule of ATP is used in the process. So net gain कितना हुआ, 2 ATP है ना, 2 molecules वो देता है, 2 molecules वो यूज़ करता है, तो लास्ट में कितना molecule of ATP हुआ, नेट गेन जिसको हम लोग बोलते हैं, that is 2 molecule of ATP is formed at the end of glycolysis process. एंड पीएफ के मॉलिक्यूल जो हम लोग बात किए थे, एंजाइम के बारे में जो बात किए थे, जो पीएफ के एंजाइम के बारे में जो बात किए थे, जिसका नाम था फॉस्फोफ्रुक्टोकाइनेज, वो जो एंजाइम है उसको हम लोग बोलते हैं इज़ अ मार्कर एंजाइम, बहुत इंपॉर्टेंट एंजाइम है. So this was all about glycolysis. In the next video we will discuss on the energetics of glycolysis, that is how much amount of molecule of the ATP is produced from one glucose molecule. Hope you like this video. Thank you.